So next, we're going to begin our study of biological macromolecules. We're going to start with our first type of macromolecule, carbohydrates. But first, I want to go through a short definition of what a macromolecule is. Okay, so we have four major classes of macromolecule. So macromolecule is a larger molecule, still based in carbon, but we're going to have four different types, each of which are going to have different properties. So some of those are going to be chemical, so they're going to react in different ways with, for example, water, and some of them may be physical, so whether they are liquids at room temperature, for example. Okay, all four of our macromolecule classes count as organic molecules because they all contain carbon, which is what an organic molecule needs to contain to be considered organic. And they may also contain hydrogen, oxygen, nitrogen, or phosphorus, depending on which macromolecule we're talking about. And as we go through each of the types, I'll highlight for you where you will expect to see those other atoms. Okay, so any macromolecule in each of these classes is going to be made up of smaller parts, which we call monomers. So we're going to have monomers, and when we link them together, we're going to end up with a larger molecule called a polymer. So that's just going to be several monomers linked together via a covalent bond. So here in this example, we see how a disaccharide, which is a carbohydrate, which is basically a sugar, how we form a polymer from two monomers. So here we see one molecule, a monomer, a second molecule, another monomer, and then we see a reaction. So in this reaction, we link these two rings together, and we can see that we've taken these little side groups, our two hydroxyl groups, and we've linked these two rings together, these two monomers together, into a polymer by taking these two functional groups and having a reaction, a chemical reaction happen here. So here we see we used to have two hydroxyl groups. Now we just have an oxygen in the center of these two covalent bonds between our monomers. We've gotten rid of one hydrogen and an oxygen and a hydrogen from the other functional group. So in order to form these covalent bonds, we did have to remove some stuff. And in this case, we removed two hydrogens and an oxygen. So what we actually did was we formed water. So we took out water in order to create this covalent bond between our monomers. Now, when we have a reaction like this, where water is removed in order to link two smaller molecules together into a bigger molecule, we call that a condensation reaction. So just like you have condensation when, say, it's hot outside and water starts to form on the outside of an icy cold water bottle, and then we call it condensation, or on your windshield, for example. Just like that, when we're removing water, when we're forming water, it's called a condensation reaction in chemistry and in biology. Okay, so just as a sort of study tool here, I've provided you a list of the four types of macromolecule that we have and the monomers we'll expect to see within each of them. So when we're talking about carbohydrates, as we are in this lecture, we'll see that our monomers are called monosaccharides. When we get to lipids, they'll be called hydrocarbons. And specifically in one of our types of lipids, we'll see the molecule glycerol and fatty acid chains. In proteins, our monomers will be amino acids. In nucleic acids, they'll be nucleotides. Then we'll have names for our bigger polymers like polysaccharides, polypeptides, or proteins, and DNA and RNA. And we'll get to those in the next couple of lectures. So on to carbohydrates. So we're going to talk first about the purpose of carbohydrates, and then we'll talk about the structure. So the real purpose of a carbohydrate is to provide short-term energy to the body. So I'm sure you've heard of carbs before, right? If you ever look at a food label, the number of carbohydrates in that food are listed. It's because it's an important thing to know about what you're eating. You want to know how much energy that food is going to provide you with. So carbohydrates specifically are found often in grains, fruits, and vegetables. Those are all going to be high in carbohydrates. And a carbohydrate chemically is going to be represented by the general formula, CH2ON. So what that means when we give you that general formula for a carbohydrate, what we're really telling you is the ratio of each of those atoms in carbohydrates. So what this means is that we're always going to have a ratio of 1 carbon, so that's the carbon here, 2 hydrogens, see 2 here, that's what this subscript 2 is here, to 1 oxygen, oxygen here. So 1 to 2 to 1, carbon to hydrogen to oxygen. So that's just our recipe for a carbohydrate. So this N here just means you can multiply that by anything. So the actual number, for example, could be 2 to 4 to 2, 3 to 6 to 3. So long as it's that ratio, doesn't matter how big the recipe is. Okay, so carbohydrate is going to be one of 3 different subtypes, each of which really just tells you how many monomers are linked together. So if we just have a monomer, if we just have 1 ring of a carbohydrate, we're going to call that a monosaccharide. If we have 2 rings linked together, as in the example for how we take a monomer to a polymer, that's actually going to be called a disaccharide. So whenever we see this word saccharide, that basically just means sugar. It's a root that means sugar. So monosaccharide, mono is 1, so 1 sugar. Disaccharide, di means 2. So disaccharide is 2 sugars. And then our big polymers. I showed you only 2 in the example, but a polymer is actually going to be many different monomers linked together. So a polysaccharide, poly means many, saccharide means sugar. So that's going to be many sugar molecules linked together. So our simplest carbohydrates are called monosaccharides. So monosaccharides are relatively small. They're going to have about 3 to 7 carbons. So linked together. Here we can see 1, 2, 3. Here we can see 1, 2, 3, 4, 5, 6. Now the 6 carbon sugar is actually glucose, which is going to be our most important carbohydrate. So if you remember any monosaccharide names, I want you to remember glucose. We'll come back to that when we get to metabolism. Most of our monosaccharides are going to end with the suffix "-ose". Each of them is also going to have an enzyme or a protein. Well, that's another macromolecule type. We'll get to that in a little bit. That breaks down that specific compound. They're usually going to end with the suffix "-ase". And so I'm just highlighting that for you here so that you don't get them confused. So if it's a molecule ending in "-ose", it's a carbohydrate. If it ends in "-ase", it's going to be a protein. Okay, so we said that our carbohydrates have this 1 to 2 to 1 ratio, carbon to hydrogen to oxygen. And part of that is because of the functional groups that are present in these carbohydrates. So here we see our 3 carbon chain, carbon, carbon, carbon. Okay, so we have our hydrogens, 1, 2, 3, 4, 5, 6, and 3 oxygens. So we know it's a carbohydrate. Now, the reason we're going to have those oxygens is really that they're going to be present in some of our functional groups that provide very specific properties to our carbohydrates. Specifically, these are going to be some polar functional groups. We can tell that because of the presence of oxygen. Remember that oxygen is highly electronegative, so it's going to carry a slight negative charge on it as it attracts extra electrons. Okay, so the functional groups that we're going to hopefully recognize in our carbohydrates are our hydroxyl groups. Now this will be the most common. So we'll see a lot of single bonds to hydrogen, but we're also going to see a lot of single bonds from a carbon to this oxygen-hydrogen combination, our hydroxyl group. We're also going to see some double bonds, carbon to oxygen. So here we see one here, carbon double bonded to oxygen. That's our carbonyl group. So we'll note here that both of these are polar molecule, or functional groups, I should say. So these polar functional groups on carbohydrates are going to give carbohydrates polar properties, which is going to be important as we think about how they interact with water. Okay, so remember when we talked about aliphatic and aromatic hydrocarbons? This is really kind of going to come into play here as we start to talk about what our monosaccharides look like. So usually we're actually going to see our monosaccharides in ring form here. So we're going to see these carbohydrates existing as a ring. But you saw on that previous slide that most of our monosaccharides, a couple slides ago, and why don't I scroll back for us? So we saw them in this linear form, right? Now what actually happens is that monosaccharides, if they're in a linear form and you drop them in water, they actually spontaneously form a ring. So we go from this linear form to a ring form, and it's the same monosaccharide. So this is ribose, but this is also ribose, okay? So these are the same molecule, just different forms of the same thing. So carbohydrates are going to have this ring structure in aqueous solutions. And that's really important because most cells are located in water. So I say that really because we're gearing up to talk about multicellular organisms, and eventually most of you are going to spend time learning about how these different cells interact in humans. Now humans are mostly made up of water. We got blood moving through us. We got interstitial fluid, so water between our tissues and cells. So when we see carbohydrates in multicellular organisms, they're always going to be in a medium that involves a lot of water. So when you try to recognize a carbohydrate, expect to see it in this linear form. So in this class, when I ask you to identify different carbohydrates, what you're going to be looking for is these rings. These rings. You're going to count the carbons, count the hydrogens, count the oxygens, and when you see that 1 to 2 to 1 ratio, it's a ring, you're going to know it's a carbohydrate. Okay. Our polysaccharides, so our polymers of carbohydrates, are just going to be long chains of these monosaccharides linked together. So we're going to see these ring forms linked together into a long chain. They're going to be linked together by something called a glycosidic linkage, which is really just a special type of covalent bond. So it's this type of covalent bond that we see between monomers of carbohydrates, and we named it special just to emphasize the type of bond. So glyco also means sugar, acidic, just like an adjective ending form. So this is basically just a sugar bond. Now, we looked at how it formed actually when we talked about condensation reactions. So if you want to see an example, scroll back to that slide. But what I want to emphasize here is that when we look at the polysaccharide, so when we look at the polymer, this glycosidic linkage is always going to look like a ring, so like our monomer, covalently bonded to an oxygen, then covalently bonded from that oxygen to the next ring, and so on. So it's always really going to look like we have a bunch of rings holding hands with an oxygen in the middle. So these are our fists or hands or something. Not doing very good social distancing, right? Okay. But that's our carbohydrates. So when you're looking at a long chain of rings, you're going to want to suspect that it could be a carbohydrate. And to confirm, you're going to count your carbons, oxygens, and hydrogens. But another really quick way to tell you're looking at a carbohydrate is to note that you have these bonds where our rings are linked together with an oxygen here in the center of those bonds. Okay, so polysaccharides can also come in multiple forms. So some of them are going to be linked all together just in one chain, We call those unbranched polysaccharides. So unbranched, just linked hand to hand to hand. We will also see branched polysaccharides, which just means that instead of bonding at this one specific location, sometimes we're going to go down, have one bond down to a monosaccharide here, and then that monosaccharide may be attached on either side itself to more monosaccharides. So this is what it looks like in its sort of chemical representation. If we drew it out as a cartoon, we might see something like this. So polysaccharides are going to be used partially for storage. So I said it's for short-term energy, and that's true, but sometimes we don't want to use glucose, for example, immediately, like right that minute. Sometimes we might want it, say, an hour later. That still counts as short-term. So we're going to have some storage polysaccharides like starch, which I'm sure you've seen in foods, right? Like potatoes are starchy, so that's a storage molecule in plants. And in humans and animals, we're going to see the molecule glycogen. So glycogen is a storage polysaccharide that you'll see in muscle tissue and in the liver that can be broken down for fast energy in humans. Now, storage polysaccharides are really focused on getting as much energy, so as many monosaccharides, as many glucose monomers, squished in as small a space as possible. So they're going to tend to be branched polysaccharides. So let's look back at the branched polysaccharides. So they're going to have these branches because we're trying to bunch together as many rings as possible. Okay, so we can see here glycogen branching, amylopectin, which is starch, branching here. We also have structural polysaccharides. So an example of a structural polysaccharide, structural means it's like for building, right? For making things structurally sound, for holding things together. So cellulose we see in plants, it makes up the cell wall of plant cells. And that's a structural polysaccharide. So it's really unbranched. So it comes in these chains. And then these chains are actually going to interact together through things like hydrogen bonds to really form a rigid structure. And that's carbohydrates.